Hi, my name is Jedward Swain. I'm currently a PhD student at Cold Spring Harbor Laboratory in New York, where I study cancer metabolism, which, broadly speaking, is the study of all of the molecules that cancer cells both consume and produce for energy and also to grow. Now, specifically, my project is focusing on brain metastatic breast cancer cells and how they interact with astrocytes, which are a cell type normally present in the brain. So brain metastatic breast cancer is breast cancer which is spread from the primary tumour in the breast and formed a new tumour in the brain. And it's already known that these cells in the brain interact with cells which are resident in the brain, such as astrocytes. But what's not so well known is how metabolites, so the small molecule exchange between these cell types, plays a role in breast cancer brain metastasis development and progression, which is what I'm aiming to study. Now, to do so, I primarily take quite a reductionist approach where I'm starting off by looking at cells in culture, with the idea being to use this model to try and identify specific metabolites which are exchanged, which I can then focus in on because they're of interest to me. And so to do so, a lot of my research takes place here, which is in the cell culture room. So if you can see behind me here, we have the incubators, which is where we keep the cancer cells and the astrocytes under very controlled conditions, so controlled temperature, humidity, and CO2 concentration to keep them happy. And if I switch the camera around and show you in front of me, you can see where I'm currently setting up an experiment in the sterile culture hood. So here in the cell culture hood, I'm setting up an experiment to look at whether astrocytes produce metabolites which cancer cells can take up and help the cancer cells mitigate stress. Now, stress is very relevant in the context of cancer, in particular oxidative stress, because cancer cells that become too stressed die. And this is actually a very important mechanism of action of a number of common anti-cancer therapies, such as a number of chemotherapies and radiotherapy, with radiotherapy being particularly relevant to the treatment of brain metastases. Now, to explain what's going on in this hood, I'm currently setting up an experiment where I have astrocytes in the flask on the left-hand side and cancer cells in the right. And behind that, you'll see the red bottles, which are different media formulations which the cells are grown in. Now, for this experiment, I'm going to first start by replating the cancer cells you see in a dish into a plate with lots of different wells. And to each of those wells, I'm going to provide a different treatment condition. So I'll have control, which is just the regular media alone. To some of the wells, I'm going to add the media, which is currently sitting with the astrocytes here. So this is called a conditioned media, and it contains everything that the astrocytes have released into it while they've been growing in it. And then the other conditions I will use is regular media with a few added metabolites, which, based on prior knowledge, I hypothesize may be driving the ability of astrocytes to reduce stress in cancer cells. So once I have all that set up, I'm going to irradiate the plate to simulate radiation therapy, and then after several days I will come back and I will count the cells to see how many died as a result of the radiotherapy and how many are still alive. Now, from previous experiments, I already know that astrocyte conditioned media reduces the stress that the cancer cells experience and so reduces the effect of the radiotherapy. What I'll be adding in this experiment is to see whether the specific metabolites I've added recapitulate that same effect. If they do, I've identified potentially a mechanism by which astrocyte-released metabolites contribute to a reduction of stress in brain metastatic breast cancer cells. Once I've demonstrated that in vitro in this model, my next step would be to try and understand the mechanism of how that exchange occurs and hopefully find a target within that pathway producing the metabolite which I can target with a drug to stop that exchange happening and thus prevent the desensitization of cancer cells to the therapy. My next step after that would be to try and test this proof of principle in a mouse model of brain metastatic breast cancer. And all going well, if that's successful, that's something I can write and publish a paper on, explaining my results, with the ultimate goal potentially that is maybe capitalized upon in human clinical trials, and perhaps this becomes a new drug treatment for brain metastatic breast cancer, whereby combining this new drug targeting this pathway I will hopefully identify with radiotherapy improves the response of these brain metastases to that radiotherapy.